Okay, hi everybody. Welcome to Bio 348. My name is Candace Orrington and I will be your TA for this semester. I just want you guys to take note of the fact that your practicals are on a Monday. Now, at this stage, we don't know if you are going to do online practicals or if you are going to do in contact practicals at the lab. Unfortunately, this is not something we could have prepared. Dr. Nati has to contact the powers that be after the semester starts. Semester starts on the 16th, which is a Monday, which means you will need a practical. And we are going to do a practical today. You do have a prac that you need to hand in. It is for marks, and we will discuss it shortly. So, regardless of whether you do online practicals versus practicals in the lab, you will have to do a practical report. You will have to write up a mini paper for each practical. And don't worry, based on whichever method or we go, whether it's contact or online, I will explain each practical, what is required of you, etc., etc. But there's a few things that I just need to discuss generally before we discuss today's practical. First things first, if you know me from Bio 204, if you know me from 212 or somewhere else, you know that I am a stickler for plagiarism. I will find you and I will catch you. That is just how it's going to go. Don't think that you can plagiarize. It's not going to happen. And I'm going to tell you what I am particularly going to instruct the demis to look for when marking your practical reports. But yes, so you will have, we will stick to this timetable of practicals almost. It will just be depending on which format we do, online versus formal. And unfortunately, like I said, we will only be able to give you instructions off once the semester's commenced. So now that the semester commences tomorrow, which is Monday the 16th, I'm recording this on the 15th. Dr. Nati will be able to put a request in for you guys to return to the lab. Whether that happens in the next few weeks or the next week, I don't know. So at the moment, we're playing it ear by ear, but you will have timely news of whether you need to come into the lab or not. Don't worry. But today's practical, which would be the 16th, is online. Okay, now for each practical that you do, you will get two options. You will either get a full practical report or a short practical report. Now, you see these percentages, just ignore them, okay? Just ignore them, just ignore them. For a full, so don't follow this on page 10 of your practical manual. Don't follow this. I want you all to jump down to page 24, I think, 22, 24, to this mark sheet. This mark sheet is how your marks are going to be divided for every single practical report. Now, remember I said you will either have a full practical report or a mini practical report. In a mini practical report, this is why I'm telling you to ignore that previous page and how it is allocated, because in a mini practical report, you need a title, you need a results section, a discussion, a reference, and then you will get this overall impression mark as well. So if it is a full practical, you will need all of this: title, abstract, introduction, methods and material, results, discussion, references, general, and all of this comes out to 100 marks. If we are doing a mini practical or a short practical report, you need a title, you need a results, you need a discussion, references, and general. Okay, and if I'm not mistaken, that should come up to 67. That should come up to a mark of 67, so out of 67. So please just follow this report, this mark sheet. Do not care about what was on that previous page. I'm telling you now. I will tell you way in advance whether it is a long practical report or if it is a short practical report. And the demonstrators and myself will have this sheet in front of us when we mark it. So when we mark your title and it is from the practical, so if you look at practical 1, which is the carbon dioxide practical, the title in your practical report is Responsive Plants to Short-Term Atmospheric CO2 Enrichment. If you have put that on your practical report, you're going to get a big fat 0. And here's the thing. It's plagiarized, which means this whole thing is 0. This whole thing is going to get a 0. I don't care about those other two. This whole thing will get a 0. If in the methods and material you have gone to a practical, for example, tissue water potential practical 3, and you've gone and copy and pasted this. You've literally just gone copy paste, which is what you think which is what a lot of you do, and you think we don't catch you. If you copy and paste that, you're going to get 0 for this entire section of methods and material. And the same thing goes with the discussion and the introduction if you have copy and pasted from papers. I will catch you, I will find you. I have one of the best plagiar sniffer outers the department has at their disposal. I will use her and she will find you. Don't do it, because here's my warning. The first offense, you will get 0 for that section. So if you plagiarize the methods and material, you'll get a 0 there for your first attempt. You will be flagged. I will flag you. The second time you plagiarize, this total mark over here will be 0. And then I will hand you over to Dr. Nati, and Dr. Nati will make the decision of whether to hand you over to sign a admission of guilt. So the decision is yours. I dare you. Try it. We will find you. I've done this too long to not be able to identify you. So that's my moaning done. Don't plagiarize, and you and I are going to have the best relationship that there ever was. Okay, so you're following this mark sheet when you're writing your reports. You're going to give a good title that is unambiguous. You know exactly what it is. It's short, it's sweet, and it's relevant to the study. Your abstract, obviously, it's giving a little bit of information. Make sure that it's balanced, that you've got the aim in there, you've got the general results and the general findings and conclusion afterwards. Your introduction. Now, I want you to pay attention to the introduction. First of all, you guys know you need background, you need synthesis, whatever. Aims and objectives, hypothesis and predictions. Now, you are at a stage where you are stepping out of Bio 200, where you had to write an alternate hypothesis, a null hypothesis, a this and a that, and a this hypothesis and this prediction. If you read papers in good journals, you will generally find that they only give aims. Sometimes they give objectives as well. Most of the time, they either choose an aim or a hypothesis. What you need to do for me in this introduction, you need to give an aim, and as long as that aim is full encompassing of the study, you can get away with it. So we're kind of grouping this together. If you want, you can do an aim and a hypothesis. That's fine if that's what you want, but it's not necessary. We are allowing you to grow and move away from this alternative and null hypothesis. The only thing I'm going to ask, if you're going to give us a hypothesis, I don't need a null hypothesis. Do not give us null hypothesis. That is nowhere. It is only in statistics. It's not for biological sciences. So in your introduction, you can make a decision whether you want to give the aims and objectives and hypothesis, or you just want to do a well-constructed research question or a well-constructed aim. It's up to you. If you are going to give us a hypothesis, do not give us a null hypothesis. Null hypothesis in the introduction is meaningless. It's for statistics only. Okay. Methods and materials, like I've said, do not copy from the Prac manual. I want to draw your attention to these variable analysis, assumptions, the statistical tests, and all of that. We are also giving you the option to do stats. You do not have to do stats. It is not a requirement. However, it will be in your benefit to do it, because if I need to find marks, we're going to find it in those statistical sections. But what is important? In the methods and materials, when you are saying the variables analyzed, you need to tell us that this is the dependent variable, this is the independent variable, this is my control, this is my treatment, this is what, you need to explain those to us. It's no point in just giving us the recipe. You need to tell us, okay, this was the independent, this was the dependent, this was my control, this was my biological replicate, this was my technical replicate if you did any. And then obviously assumptions of statistical tests described adequately. Now, if you are going to do stats, you will obviously put all of that in there, as well as your Excel data. The stuff was generated on an Excel graph sheet. If you are not going to do stats, you run a very high risk of not getting full marks for everything, but the decision is completely yours. But still, for the marks for statistical analysis and assumptions of a statistical test, if you have chosen not to do statistics, we will mark you based on your ability to say Excel was used to generate the graphs. And also remember that mean and standard deviations are also a form of statistical analysis. Okay, now the results section is what we're going to focus on in today's practical, and then obviously the discussion and conclusion. A lot of please remember, you've given an aim in you've given an aim, you've given a hypothesis, you've given a prediction, or whatever the case may be. You need to confirm or deny it. Even if you say in your discussion, the aim of this experiment was, or the aim of the study was, it was found that is, you are accepting or rejecting. But you need to be able to explain that you know what your aim is, you know what your hypothesis was, and you are accepting it or you are rejecting it, or you managed to obtain or reach your aim. Students tend to completely forget to compare and relate results to other studies, and we're going to kind of look at that today as well. And then this soundly address the biological implications. Okay, you found this out. What does it mean in terms of the bigger picture? And please, suggestions for future work. Repeat the experiments is not a suggestion for future work. I beg of you, do not do the experiment again. We will mark, you will get a 0 for this little 2. You will get a 0. Suggestions for future works are things like, you know what, we measured all of these, we didn't measure this. This is beneficial to measure because X, Y, and Z measured it as well, and it was beneficial. You know, this parameter is closely linked to this parameter. We should have done it. This should be looked at. You know, instead of just looking at temperature, we should look at salinity as well. Things like that. That is suggestions for future work, not redoing the experiment or increasing the rep numbers. That is garbage. That is useless. It is stupid. If you have to tell people that you needed to increase your replicate numbers in suggestions of future work, you didn't do a good enough job in the beginning. So please scratch that out. That is not useful suggestions for future work. Now, references. Let me show you actually. I'm on YouTube. Okay. We are using the South African Journal of Journal of Botany. So you come here, you'll see South African Journal of Botany. You'll click Guide to Authors. Yes, we accept all cookies. Download guide. Okay, so then you get this thing. Now, we, for this module, are following the South African Journal of Botany. So you're going to scroll all the way to the bottom. If you were really smart, you would follow all of this information. It tells you what size, it tells you what type font, how you write the taxonomy things, everything like that. Examples of specimens, it tells you everything. It tells you how you should do your tables, figure captions, JPEGs, all of that. But this is where I really want you guys to focus, is the references. We are following the South African Journal of Botany, and it gives you examples right here. It gives you examples to follow. It shows you how to reference, web reference, data references, references from management, referencing style, single author, two authors. You guys need to go through this. A lot of students pass and fail on this references. It's 5 marks, but for some reason, it's almost like the references decide whether you fail or pass that practical. I've shown you now, you go to the South African Journal of Botany, we're following their referencing style, and you literally just do it like they've done it in the examples. Now, and then obviously a general overall impression. You know, how did I feel reading this? Did it make me want to cry? 0. You know what? I actually saw that the student put a lot of effort into it, and it was an enjoyable read. 5. You know what? The student tried, but still made me want to cry. 2.5. So, you know, and also, so this is your marking sheet. When you are print this out. When you are writing your practical reports, follow this. If you have done every single thing that this document, this little piece of paper has told you, you will get your marks and you will pass. Please just remember that for a short practical report, I want title, results, discussion, and conclusion, references. For a full, everything. Okay. Now, graphical representation of data. This explains to you what's independent, the dependent. You should know all of this from Bio 200. So you can go through this before you jump to your practical for today, which will be uploaded. Here's your practical here. Okay. For 50 marks, you will have to upload it. It is your practical 1. Okay. I have given you a set of raw data. I've told you that Amaranthus dubius, which is an African leafy vegetable, it is also used to make marog, was germinated in petri dishes on germination paper or filter paper, it didn't matter. And we watered it with water supplemented with increasing concentrations of salinity, salt. So we started at 0. So we've got 1, 2, 3, 4, 5, 6 concentrations. This 0 concentration, the 0 millimolar, will be our control. Okay? So that 0 is our control. So, and we've got 1, 2, 3, 4, 5 treatments. Now, germination and seedling emergence was recorded at the end of 28 days, and you must take note that there were 10 biological replicates, and we did it 3 times. So 1, 2, 3, and in each one of these reps, we had 10 individuals. And I took germination when it produced a radical that was bigger than 1 centimeter, and I took seedling emergence as a full seedling with 2 cotyledons. If all of this is unfamiliar for you, it should be familiar, because you had to do Bio 204 in order to do this module. Just go, I suppose I could show you as well. I've got Google open, so if we type in a radical, okay, this little thing. Okay, so here we go. My internet is slow. So if you look here, this is the radical over here. That's the radical over there. I'll just ignore that now. Here we go. There's the radical over there. So that's what I took as, so this had to be greater than 1 centimeter to count as germination. And I needed a full seedling with 2 cotyledons, so these bottom leaves first, or to be, Amaranthus is a bit different. This looks like a common bean plant. Amaranthus is a little bit different. The true leaves are the cotyledons in the first place. And I'm going to sneeze, and I'm not pausing, so excuse me. Sorry. Okay, so when I say, sorry about that, guys. When I say the radical was greater than 1 centimeter, I'm referring to this. But that, just because you germinated doesn't mean you're going to grow to a full seedling. Okay? So I needed germination and I needed seedling emergence. So seedling emergence. And what you should notice very clearly is that just because I germinated on, let's say, 100 millimolars doesn't mean I got a seedling on 100 millimolars. All right. Now, I've given you this information. And when you look at this, you can see here that out of my 10 on the 0, which is the control, I got 10 germinating. So all 10 germinated. 2 didn't germinate in rep 1 here, 1 didn't germinate here, and 2 didn't germinate here. So you need to decide, and I'm going to give you a hint, and I'm not putting it on here because I wanted you guys to be able to listen to this whole thing. The hint is the best way to do germination and seedling emergence on a graph is in percentages. Okay? You still need a standard deviation and you still need an average, but show it in the graphs as percentages. Okay, so you need to take this information. You need to go and you need to look at the back of the practical manual where it gives you examples on how to do all this. And you need to take that marking sheet, your marking sheet for the results section, and you need to do a results section for this first one. So 25 marks is you need to have adequate summary of the results. And I'm going to just tell you briefly, at the end of the day, what has happened is with increasing concentrations of salinity, we got a decrease in germination and a decrease in seedling emergence. And also you need to note that as the concentrations increased, just because we got germination does not mean we got seedling emergence. Okay, so you might want to, so you need to be able to think there, okay, so this is what I should be getting. So you need to be able to adequately do a summary of the results because results are not just the graphs, you have to do a paragraph underneath. Yeah, you remember that? Okay, so that's for 5 marks. Did you represent the data in the most appropriate way? For example, we need averages, we need standard deviations, I'm telling you now that germination and seedling emergence should be done in percentages. Have you given it a descriptive title? Are you using the correct calculations? Did you do averages? Did you do standard deviations? Can I tell the difference between which one's germination and which one's seedling emergence? Did you do the independent and the dependent on the correct axes? And is the data analysis complete and thorough? So in that data analysis, remember you need your averages, your standard deviations. Don't forget you must be able to put standard deviations into your things. If you don't know how to do standard deviations, you have the whole week to contact me to find out about how to do a standard deviation, but you should not. Okay, so that's what you need to do. You need to look at your practical report and you need to give me a results section for 25 marks from the results that I've given you for the first one. Then the second one, I'm uploading this paper by Daly et al. 2021. And you need to answer the following questions for 25 marks. Okay. Now you've just done a results section. This is raw data, by the way, raw data. Now you need to go to this paper and you need to tell me if you think that they represented their data in a clear, appropriate way. So essentially, you take that marking sheet and you have a look and you see, you know what, is the data presented in the most appropriate and clearest way? Can I understand what these authors are saying? Are these results too confusing? Does it look like it should look in a journal? And then you need to look at what it means to be ISI accredited. Now, us as a university, ISI accreditation good, no ISI accreditation bad. ISI accreditation essentially means that now don't copy, I know what I'm saying, you need to go and do proper research, but essentially, let's say I publish a paper in a journal that is ISI accredited. It means that the university has acknowledged that this journal is a good journal, and an independent body of ISI has accredited this journal to say it is peer-reviewed, it goes through the correct channels, and it is done appropriately, but most importantly, it is peer-reviewed. So you need to have a look at this journal Journal of Medicinal Plants and Studies, and they even give their website, and you need to tell me if this journal is accredited. And you need to tell me how you figured that out and how you did not. And it's very easy. Just type in ISI accreditation and the journal name, and you should see something very scary. Has to do with a predator. Hint, hint, wink, wink, nudge, nudge. So you need to explain to me why this journal is or is not ISI accredited. And then I'm asking you, should you as a student use a non-ISI accredited paper when writing a report or paper? I'm going to give you your 2 marks there. No. Absolutely not. Because if it is not ISI accredited, it could be a predatory journal. And predatory journals are not peer reviewed, which means it has not gone through the proper process of being reviewed, which means there could be so many mistakes in it. It's not scientifically correct and accurate. There's no backbone to it. There's no secure knowledge in the fact that this is a good journal. Okay, so then obviously, what is a predatory journal? So if you are not ISI accredited, you could just be waiting for your ISI accreditation, but you could also be what is known as a predatory journal. And predatory journals have a high turnover rate, which is something that a lot of scientists want. They don't want. I mean, it takes, we just looked at the Journal Botany of Science, the guide's authors. It could take 3 to 4 months just to get accepted before your paper is even published. But it's peer reviewed, it's an ISI accredited journal. Exactly. So then ultimately, should you use Daly et al. 2021's paper for your paper? And you should, by my hinting and winking and nudging, already know what the answer to that is. And just having a look at Daly, as opposed to the example that is given in your book from the South African Journal of Botany, you should be able to figure it out. But I just want to show you some things that should also, just send a red flag into your brain. From referencing, if you are referencing this style, why are you putting that style there? Why are you chopping and changing between styles? What's the point? What's the point? I don't understand. Now, this is something that I picked up, which I'm not expecting you to be able to pick up, but chlorophyll and carotenoid content, this technique is very old, very outdated. Hiscox and Iselstrom is not widely used at all. We tend to focus on the Aaron paper for carotenoids and uh, chlorophyll content. And this was a red flag to me, but this is my field. So I would never have expected you guys to pick that up. I'm just telling you, it's my field. So I was able to pick it up. Ultimately, I couldn't figure out what was going on here. If you could, you deserve a medal. I couldn't figure out what was going on here. It's a mess. It's awful. And then look in that reference list. That is not how you reference. That is awful. What do you mean et al? You've given the list of the names and you're putting et al. Et al means that there's more. You need to put all in a reference list. So just from that, you should be able to tell what kind of journal this is. Just go and confirm it. And I'm completely okay with you guys screenshotting websites to help support your answer, but then just explain what that, don't just put a screenshot in there. Just say, look, I went onto the internet, I found this website which told me this about this journal. Okay, so you need to answer these questions for 25 marks. You need to give me a proper results section using your marking sheet, and you can hand it in. You can answer it on here, put your name, student number, and re-upload it onto Moodle. I am hoping that this will go off without a hitch tomorrow, which means I would expect it in a week, which means hand-in will be in on the 23rd of August. However, I will keep you posted. If I can't upload it, I will change the date in the instructions when it is on Learn. All right. Wish you the best of luck. And please, oh, right. I will just quickly put, I'll upload it onto Learn as well. But this is my email address. You can expect communication during normal working hours between 8 and 4, Monday to Friday. You cannot SMS me or email me on a Sunday because something's due on a Monday and expect me to respond. It's not going to happen. It might, but it's probably not because I'm napping on a Sunday. So if you need me, you can contact me. We are here for you during the week. I will answer any questions. If I have to upload tutorials on explaining things, I will. But unfortunately, I cannot see your beautiful faces. I can only guess that you've understood what I've said and that everything's going to be okay. So if you are battling and you are struggling, I don't know. I can't see your facial expressions. I don't know. Speak up. I will help you. That is what I'm here for. But I will do it during a normal work time, please. All right. Good luck. Uh, please email me if there is a problem, and I will try and sort it out. But yes, this is your first practical. Good luck. Okay. Goodbye. Stay safe. So much stuff.